that broke down sugar? Or was there some other way that mitochondria and chloroplasts ended up as part of a cell? And what scientists put together is called endosymbiotic theory. Endo meaning inside, and if you're familiar with symbiosis, symbiotic is just a relationship. So living in symbiosis is having a relationship. Endosymbiotic theory basically says that the way that mitochondria and chloroplasts came about is that you had one big prokaryotic cell that was maybe more complex. It ate a smaller prokaryotic cell. This prokaryotic cell happened to be one that could break down sugar with oxygen, basically what mitochondria do. Over time, over many, many years, basically that mitochondria lived in the cell and they had a symbiotic relationship, and eventually it lost the ability to live on its own and it just became an organelle that's part of the cell. Which sounds kind of crazy. You might say, that just sounds stupid. There's actually some evidence, which I'm going to show you on the next slide, to support it. Why do scientists think that this makes more sense, that this is something that got engulfed millions of years ago and eventually just became part of the cell, as opposed to that the cell somehow manufactured a mitochondria over millions of years? Again, it's not just mitochondria, but also chloroplasts. So these are the two organelles that endosymbiotic theory is related to. That the photosynthetic cell that lived on its own, that was a bacteria, was engulfed, and that eventually then became a chloroplast. All right, so let's talk about how they come up with this. Why do they think this? This is just a diagram showing like this ancestral cell that a nucleus was starting to form. It engulfed this thing that eventually became a mitochondria. And some of them also engulfed this other prokaryotic thing that was photosynthetic, and eventually it became a chloroplast. All right, evidence. So why do they think that this makes more sense than them making it on their own? Mitochondria and chloroplasts, number one, have a double membrane. Now, it's not just that they have a double membrane. In other words, if a cell eats something, here's what happens. This is the cell. This is the thing that it's going to eat. What's going to happen is that thing that it eats is going to end up with a piece of cell membrane around it. In other words, it's going to have its own membrane, and then it's going to have a second membrane that came from the cell that ate it. Now, not only do mitochondria and chloroplasts both have this, a double membrane, the inner membrane that they have is very much like what bacteria have. It's a bacterial type cell membrane. And the outer membrane is more like what eukaryotic cells have. So if this is true, if this is what happened, this piece of evidence supports it. That a cell ate another cell, it would end up with a double membrane. Its own inner membrane that it started with, and then a second membrane from the piece of cell membrane that surrounded it. So that's piece of evidence number one. Piece of evidence number two, they have their own DNA and ribosomes. If they were indeed a bacteria that lived on their own, at some point, they would have needed to show the characteristics of living things, one of which is having their own DNA, their own ribosomes, and their own metabolism. Mitochondria and chloroplasts have their own DNA and ribosomes. Not only do they have their own DNA and ribosomes, but their DNA is like a bacteria's DNA. It's a loop of DNA like a bacteria. And their ribosomes are also bacterial ribosomes. I don't know if I mentioned to you that bacteria's ribosomes are smaller than eukaryotic cell ribosomes. So their DNA and ribosomes are bacterial in nature, and the nucleus doesn't control them. They actually reproduce themselves. So when the cell divides, the mitochondria then turn around and divide also. Now, they can't survive on their own. They're not living things. You can't take a mitochondria out and let it go and it becomes its own cell. It's a cell part. But this evidence does support the idea that maybe millions of years ago, these were some kind of bacterial cells that lived on their own. They were engulfed by another cell. Instead of getting digested, they lived in a symbiotic nature. The cell reproduced, they reproduced, and over time they basically just lost their ability to live on their own, and they became a permanent cell part. So that's the evidence to support endosymbiotic theory, and you should just be familiar with what the theory says and what the evidence is that supports it. Yes? Yeah, and that the DNA and the ribosomes in a eukaryotic organism look different than that in a bacteria, and the DNA that's in the mitochondria and chloroplasts look like what you see in bacteria, not like what you see in typical eukaryotic organisms. All right, so that's endosymbiotic theory. The very last thing we're going to run through very, very quickly is the cytoskeleton. So when I was in school, I remember learning that the cytoplasm was this jelly-like stuff, and all the organelles floated around in the cytoplasm. In fact, that may be how you envision it. That when you think of a cell, you think of the ribosomes and all that stuff, and there's a jelly, and they're all just sort of floating around in the jelly. That's not the case. That would be the same as saying that your lungs and your heart and all of that stuff is just floating around in your chest and abdomen. So you're jogging and it's bouncing all over the place, and that's not what's happening. You know that your lungs and your heart and all that stuff are held in place. It turns out that the parts of the cell are also held in place. They're not free floating around in a jelly. So the cytoskeleton is what does that. There's three components. The first one is called microtubules. Easiest thing to remember about microtubules, they're made of tubulin. Tubules, tubulin. That will be easy to match up. They are actually the biggest of all the parts of the cytoskeleton. Specifically, what their jobs are, number one, they give cells their shapes. So cells are not amorphous blobs. They actually do hold specific shapes. Muscle cells look a lot different than nerve cells or bone cells or blood cells. And those cells do have a shape. And the microtubules are the main component that does that. Secondly, cells that have cilia and flagella, the microtubules are what compose cilia and flagella. Now, in us, I know there's cilia in our respiratory tract. Like not our cells, not all of our cells have cilia. And the only cell in humans that have flagella would be sperm cells. But in other organisms, there are cells that have cilia and flagella. Some single-celled organisms move only by flagella or by cilia. There's a lot of different jobs that they do. So in us, we don't necessarily see lots of cells with flagella. But that is what those are made of. Also, chromosome movement. When I say chromosome movement, just to comment on that, I don't know how well you remember having to learn mitosis. But there were these things that went across that pulled the chromosomes apart. They were called spindle fibers. That's what they're talking about. Those fibers that pull the chromosomes apart are made of microtubules. And these little things on the ends, these were called centrioles. They look kind of like little T-shapes. Those are also made out of microtubules. So those are sort of the main things that we see microtubules doing in a cell. And when organelles do move around, microtubules do control where the organelles go. So nothing is really free-floating. Everything is very controlled where these organelles and things are moving in the cell. So that's microtubules. They're the biggest. They are actually, I know I listed sizes here. You would not be tested on the sizes. About 25 nanometer diameter for them. Microfilaments is our second one. Oh, but I think before that, this is what these are centrioles. Again, not on a test anywhere. And this one, so if you were to take a sperm and cut its tail off, which would be very cruel, but if you did, this is what a cross section would look like. So these are actually, it's made out of a whole ring of like 9 sets of microtubules and 2 more in the middle. So it's actually really complicated, their tail. It's not one little strand. Under a microscope, it's a bunch of microtubules that give it the power to move. So that's what microtubules look like. The second one, microfilaments. Microfilaments are made out of a different protein called actin, which is not as easy to remember because tubulin and tubule went together. Actin and filament, no. The thing about these are the smallest, only about 7 nanometers. Again, you wouldn't be tested on their size. They are very strong, so they can bear tension. They are involved in the cleavage of cells, and by cleavage, just to clarify the meaning of that word, I'm talking about cells pinching apart when they split during like mitosis. They actually pinch and pull the cells apart when the cells divide. White blood cells actually have these pseudopods where they actually can extend and surround things. That is controlled by them. But I would say, and the reason I put it in bold, is that on a test, the one function I would ask you would be this one. Microfilaments are what allow your muscles to contract. So if you took anatomy and you remember learning about the sliding filament model, microfilaments are in your cells and they slide past each other, and this is what allows all your muscles to contract. So cell cleavage and some of these other things They're the smallest ones. They're made of actin, but muscle contraction would be the one function. If I was going to ask you, that would be the one I would want you to remember. The last one, just to wrap it up, intermediate filaments, I probably wouldn't ask you anything about them, other than that they're in between. They're kind of more permanent. They hold things together. They're kind of in the middle in size. And that's pretty much it on intermediate filaments.